welcome everyone to today's CMR German Club, also on behalf of Matthias Friedrich, my co-host. And happy New Year to all of you, as we already said. So today we have two, as I find, very interesting papers, as always. Actually, this time we will cover atrial strain analysis, and we have two different papers. One is a bit more technical or methodological on reproducibility of atrial strain analysis. And this paper will be presented by Anvesha Singh from Leicester. And the second paper is on atrial strain in patients with atrial fibrillation and LGE. And this is a work which will be presented by Luuk Hopman from Amsterdam. So welcome to both of you, and thank you that you present your papers today. So I think we start with the first one, which is entitled Reproducibility of Left Atrial Function Using Cardiac Magnetic Resonance Imaging. And you can share your screen, and the floor is yours.
Can you see that?
Yes.
Great, thank you. So thank you so much for the invitation to present our paper. We're delighted to do that today. Our paper, as Bettina mentioned, is about reproducibility of left atrial function assessment using MRI. So before I begin, just a very brief introduction to the topic. Why is this important? So we know that left atrial remodeling has been associated with diastolic dysfunction and is a well-used imaging biomarker in many cardiovascular conditions. It's been shown to predict adverse outcomes across a range of cardiovascular diseases, including aortic stenosis, type 2 diabetes, chronic kidney disease, and heart failure with preserved ejection fraction. But more recently, there's been a lot more focus on LA strain, which is emerging as a novel imaging marker. And this is exciting as it has the potential to overcome some of the limitations of volumetric assessment. It can also provide potentially early detection of abnormalities even before the volumes increase in some condition. And early papers have also suggested that it does have prognostic value in some conditions. But before we can routinely use any imaging biomarker in clinical practice, we need to establish its reproducibility. And whilst many papers do report inter-observer and intra-observer variability of imaging techniques routinely, true test-retest reproducibility is rarely reported. And this is important because in addition to variability, it also accounts for other sources of variability that can occur in the real world, such as physiological variations in a patient from different days, radiography variability, and the way that things are planned. And this is important if we are to utilize a new technique in order to monitor disease progression or even response to treatment. So with this in mind, we set out to assess the test-retest repeatability or reproducibility of CMR-derived LA function. In particular, we want to look at LA strain and strain rate, but also the routine LA volume and emptying fraction for comparison. We did this by recruiting 60 participants with a range of cardiovascular and non-cardiovascular diseases. Majority of the patients had disease, so including aortic stenosis, type 2 diabetes, end-stage renal disease, and we also had 6 healthy volunteers. All of these 60 participants underwent 2 cardiac MRI scans, 7 to 14 days apart on the same scanner. The majority was scanned on a 3 Tesla scanner, but 17 was scanned on 1.5, but each individual patient was scanned on the same scanner twice. Of note, all of these participants were in sinus rhythm at the time of both their scans, which allowed retrospective ECG gating to be used in all scans. When it came to CMR analysis, it was done in a blinded fashion by a single observer. And by that, we mean that all images, all MRI scans were given a unique code, a 3-digit code, and the patient analyzing was blinded to both the disease condition, but also whether this was scan 1 or scan 2. In terms of analysis for the volumes and emptying fraction, we used the biplane area length method, and the software we used was QMass version 8.1, which is a MEDIS software. We did these measurements on 2 and 4 chamber cine images, which is shown in this figure here, just an example at end diastole and end systole to show you the volumes. And this allowed us to calculate the maximum and minimum LA volumes. And we were also able to calculate the LA preatrial contraction volume to give us the emptying fraction at the 3 phases of left atrial function, which is reservoir, conduit, as well as the booster pump. For the purposes of LA volume, we did exclude the LA appendage as well as the pulmonary veins. For the LA strain and strain rate assessment, we use feature tracking using QStrain version 2.0, which is also a MEDIS software. Again, we analyzed this on 2 and 4 chamber cine images, and for comparison, we excluded the left atrial appendage and pulmonary veins again. The strain and strain rate were also extracted at the 3 phases that I've mentioned before. And this is an example strain curve in figure 2, which shows how we extracted the different parts of the LA function. In terms of the statistical analysis, we use the Bland-Altman method for the reproducibility, but we also displayed the ICC and coefficient of variance in our results. For the Bland-Altman plots, the differences are expressed as a percentage of the mean, just to make it easier for comparison. So moving on to the results. The first important point was that the overall time taken for the volumetric assessment was about half that of the strain assessment. So the mean average time was about 6 minutes for volumetric and emptying fraction. The actual contours took about 9.5 minutes for the strain curves, but then there was an additional 6 minutes to actually extract the different phasic function. And this is why the total time was around 15 minutes. This study was not designed to compare the different disease groups, and therefore the patients were not matched, and therefore I'm not going to dwell on these results, but it is available for anybody who is interested. The main purpose, of course, was to look at the test-retest reproducibility. So we'll move on to that. So looking at the test-retest reproducibility of the volumes and EF, overall we found this was good to moderate for all participants. And the results here are displayed here in table 2. So here on the left is the volumes, and then the last one is the total emptying fraction, which is displayed. And these are the Bland-Altman limits of agreement, the coefficient of variance, and ICC. And overall, you can see that the COVs were around sort of 20 to 25%, where the ICCs were 0.8 to 0.9. In comparison, the same values for the strain and strain rate parameters. And again, here on the left are the top 3 are the LA strain at the different phases, and then the strain rate at the different phases. And again, if we look at the coefficient of variance, we can see these are higher, and the ICCs are lower compared to LA volumetric parameters, suggesting that the reproducibility was not as good for the strain parameters. These are some Bland-Altman plots demonstrating the same thing. So if we look at the top 2, this is LA strain at the reservoir function, and this is its corresponding emptying fraction, which is the total emptying fraction. And you can see that the limits of agreement are narrower for the emptying fraction, suggesting slightly better reproducibility. On the left here, these 3 are LA strain at the different phases. And what this is showing is that the reservoir phase was the most reproducible out of the 3 phases. Out of interest, we also looked at the correlations between the volumetric assessment and the strain to see if there was a correlation. And we found there were significant correlations at all phases, but this was poor and quite a lot of variation for the conduit and the booster pump phase, but better for the reservoir phase. So overall, our results are suggesting that the volumetric assessment using biplane area length method appears to be more reproducible compared to the novel LA strain assessment at present. Before I finish, just a few limitations to address. We know this is still a relatively small and heterogeneous group, although it is the biggest of its type to have this assessment completed. We only use the MEDIS software, so of course the results may not apply to other software and they should be looked at separately. And finally, we use both 1.5 and 3 Tesla field strengths. So some of the differences in variability may be due to inherent differences between the field strengths. So in conclusion, we have shown that the test-retest reproducibility of LA strain and LA strain rate by CMR using feature tracking is only moderate to poor across disease states, whereas for LA volume and emptying fraction appears to be more reproducible and quicker to analyze at this stage. So before this technique can be routinely utilized in clinical practice, further development of strain quantification and standardization of acquisition and analysis is needed. Thank you.
Thank you very much, Anvesha, for this great presentation. I think this is a really important study because when we really aim to translate strain imaging to clinical practice based on feature tracking, which we all aim for, I guess, at least I do, these are exactly the problems we are facing currently. And poor reproducibility or moderate reproducibility even might really pose some problems here. I am wondering, so as far as I understood, you did the test-retest sets 1 to 2 weeks apart. So might it be possible, because you investigated patients with a diseased heart, that there might also be differences introduced and thus biasing the study results due to hemodynamic changes, because this might, since strain is quite sensitive to those changes, might also alter strain values? How did you correct for this, or yeah?
Yeah, thank you. So the patient groups that we use, I'll just go back to that section just for clarity. Sorry, takes a long time to find the right bit. Yeah, here we are. So the patients groups that we did, as I said, was aortic stenosis, type 2 diabetes. So those I don't think are likely to show significant changes in their disease process within a 7 to 14 days period. But you're right that the end-stage renal disease group on hemodialysis may have some fluid changes in that period. So to account for that, we did try and, I think it is in there somewhere, we tried to, here it is, that we tried to standardize their volume status. So they were deliberately scanned on their non-dialysis day and at the same time of their kind of dialysis pattern to try and standardize the volume status as much as possible. But other than that, the general variability in hemodynamics is kind of what we wanted to capture, because that's the whole purpose, is that, you know, in real world, if you are going to monitor a disease progression or LA strain progression, there will be some variation in the you know, overall hemodynamics of the patient. So we want to include that in the variability assessment.
Yeah, I fully agree. So we want to find out which strain changes are really indicating disease and which are not, which are only indicating small hemodynamic changes, which might be normal. Yeah, I fully agree. But there's always some cutoff and potential bias in these data, which we cannot correct completely, I guess. Since there are no questions so far in the chat, another question. What is your feeling? You said that other software solutions might deliver different results. Do you have a feeling on that issue? Did you try other software versions? Because I did so, and I found quite large differences between them, but I would be interested if you also did so.
So this is something we are looking at, not so much for LA strain so far, but we have looked at LV strain with different software. And there are some differences. And interestingly, we've also found some differences between 1.5T and 3T for LV strain reproducibility. So that is a paper our group is currently sort of preparing for publication. We do intend to also look at it for LA strain in a larger cohort once we have the cohort available. But I do think there is differences between software because of the inherent the software itself, the way the contours are propagated are slightly different. So for example, tissue tracking on Circle is slightly different to feature tracking on MEDIS. So I suspect there will be differences, but I don't think there's been any studies directly comparing them to sort of say which is the least or most reproducible, unless you have that data. You said you've looked at that as well.
Yeah, it's quite difficult because MEDIS, as far as I know, is the only one providing an atrial strain solution. So if you use the other ones, you have to do it off-label, kind of, and use the RV, the LV, whatever tool is available, and that's even more time-consuming than what you have shown here.
Yeah, that's why we chose to use MEDIS, because we couldn't find any others that had a dedicated LA strain tool available.
Yeah, that's true. So one last question from the chat. From Dana Peters. Is it the long axis view that changes? So due to foreshortening,
So do you mean in terms of planning of the long axis?
Maybe you can also unmute yourself and elaborate a bit further your question, Dana. Okay, we cannot hear anything. So is it the choice of the long axis view?
So I'm not sure if I've understood it correctly, but I think what you're asking is, can it be foreshortened and therefore affect the strain measurement? Yes, the reproducibility. So that is, of course, one of the parameters. So that's what I meant when the radiographer will be different on a different day, and they will plan it slightly differently. Although, like every other center, we have very strict ways of planning to try and avoid that foreshortening. So most of our radiographers are very well trained to avoid foreshortening, but that's also part of the error that we want to capture. So we're not trying to make the scans identical because that is not possible in the real world. What we want to know is how much of the reproducibility is because of those variations and how much is it actually the disease causing the changes, as you said.
Yeah, thank you very much. So really interesting discussion and great paper. So I'm looking forward what is coming next from your group on this topic. Thank you very much again. And I think in the interest of time, we move on to the next paper, which is entitled Impaired Left Atrial Reservoir and Conduit Strain in Patients with Atrial Fibrillation and Extensive Left Atrial Fibrosis. Luuk, we can see your paper. The floor is yours.
Perfect. Thank you for the invitation. We have recently published an article in CMR, which I will discuss.
Sorry, could you make it perhaps a bit larger because it's quite small,
yeah,
at least for me.
Let's see if I can. Like this?
Yeah, perfect. Thank you.
Okay. As you may know, atrial remodeling is an important hallmark feature of atrial fibrillation. And in this regard, detailed characterization of atrial remodeling and understanding of the interplay between structural remodeling and functional remodeling is essential. For the evaluation of this atrial remodeling process, CMR has emerged as an important imaging modality, as it can be used for the non-invasive assessment of left atrial fibrosis through 3D LGE imaging, and for the assessment of left atrial function through feature tracking strain. The assessment of left atrial function is complex, as it includes three phases: the reservoir phase, the conduit phase, and the contractile phase. And CMR feature tracking can help for the accurate evaluation of these three phases by following the atrial wall over time. So in this study, we investigated the association between left atrial fibrosis and left atrial strain in all three phases in patients with atrial fibrillation. We included 105 AF patients who were scheduled to undergo their first AF ablation procedure. And prior to the ablation procedure, these patients underwent CMR imaging. And important to note is that all patients were in sinus rhythm during the CMR scan. We also included 19 healthy volunteers who served as controls for the volume and function assessment. The CMR protocol consisted of cine imaging in a 2 and a 4 chamber view, a 3D contrast enhanced MRA, and 3D high resolution LGE imaging. The 2 chamber and 4 chamber cine images were used for left atrial strain analysis using the feature tracking module in Circle CVI. Let's go to the figure. The longitudinal strain measurements were subdivided into left atrial reservoir strain, conduit strain, and the contractile strain, as illustrated over here in figure 1. see. Quantification of left atrial fibrosis was performed in open source software CMR, and left atrial fibrosis was calculated using a default image intensity ratio threshold of 1.2. Now onto the results. Good quality cine images were available in 94 out of 105 patients, and good quality 3D LGE images were available in 82 patients. We found that, go to the image. We found that in comparison to controls, all measures of phasic strain, so the reservoir strain, conduit strain, and also the contractile strain were impaired in patients with atrial fibrillation, as is shown over here in this figure and also in this table. Thereafter, we divided the AF patients into two groups according to the median percentage of left atrial fibrosis. And we found that passive strain parameters, but also passive left atrial emptying fraction, so the left atrial reservoir and conduit strain, were both significantly impaired in patients with a high degree of left atrial fibrosis, while contractile strain on the other hand was comparable between the two groups. So in conclusion, we've shown that all components of left atrial strain were impaired in AF patients as compared to same aged healthy volunteers, and a lower left atrial reservoir and conduit function was found in AF patients with extensive left atrial fibrosis. A possible explanation for this is that left atrial wall fibrosis contributes to an impaired passive left atrial function, possibly because the interstitial atrial fibrosis causes regional variation in the myocardial structure, which may impact atrial compliance. And because cardiomyocytes are not replaced by fibrosis, the left atrial contractile function remains largely unaffected. An alternative explanation may be that the contractile function serves as a compensatory mechanism to maintain proper left ventricular filling, and therefore is not different between the two groups. Lastly, I would like to point out two limitations, and that is that there's currently no 3D LGE post-processing protocol which is widely validated, and so there's no complete agreement on thresholding values to indicate left atrial fibrosis. And second, the atrial wall is rather thin relative to the LGE CMR voxel size. So it could cause measurement errors introduced by the partial volume effect. That was an overview of our paper, and I'm happy to take any questions.
Thank you very much for your excellent presentation and the interesting paper. Where would you personally see the clinical value of LA strain in these patients? So usually we say strain is indicator for subclinical disease or more sensitive than whatever, than LVEF or other measures of contractile function. So where would you see here the value in this special patient cohort?
I think it's especially for these AF patients. I think it's important to get some kind of overview of their left atrial remodeling status. So this left atrial strain, and then also the three different phases could give an could give some kind of marker what the status of the remodeling in the patient is, and possibly in the future we can identify high risk patients or patients who have a high risk of AF recurrence after an ablation procedure. So maybe we can some kind of risk stratify these patients based on their left atrial strain values.
So we can see strain as an additive parameter to LGE and other volumetric parameters or whatever. Yeah, so this prognostic value would be, I think, very interesting, because this is an unsolved clinical problem. Actually, we cannot predict who will suffer from relapse of atrial fibrillation after ablation and who will not, at least not in a very sophisticated manner currently. Yeah, very interesting. So what would you think when it comes to temporal resolution of the cine images? We know that there might be large variation of strain values. If we vary our temporal resolution, do you think we could be better than now if we would bring temporal resolution down to 10 milliseconds or so, like an echo?
That's something we didn't test in this research. I think the higher the temporal resolution, the better. But I think that's something that has to be further researched in the future.
Yeah, would be interesting as well, I think. And what was your experience with using Circle for LA strain analysis? Was it easy to use because it was off-label, I guess?
Yeah,
did you
indeed.
Use the LV tool, or what? How did you perform?
Yes, we used the LV feature tracking module. So we had to draw the endocardial and epicardial contour in the left atrium, and that's not that easy sometimes because of there's no atrial wall in the when the pulmonary veins come in and the left atrial appendage. So the feature tracking algorithm has some difficulties following the kind of non-existing left atrial wall at those points. So maybe an alternative left atrial strain calculation that is less dependent on following the left atrial wall over time would be beneficial, because sometimes it is quite difficult to for the algorithm to follow the left
that's
atrial
also
wall.
my experience, even with other software tools using a different tracking algorithm. It's always the problem with the pulmonary veins, especially if you have a heavy inflow artifacts. Then the algorithm usually tracks this inflow phenomenon rather than the atrial wall. So yeah. So there is one last question in the chat, again from Dana Peters. She says, I think the LGE enhancement cutoff of 1.2 might include too much normal tissue, but I agree that the optimal cutoff is not agreed upon and is probably patient specific. Did you look at other cutoffs?
We looked into that, and we have a paper focusing on that that is currently or is recently published in European Heart Journal Cardiovascular Imaging, because there are quite some variations in thresholding and the cutoff strategy. So you also have the strategy that you pick 3 standard deviations above the blood pool value, and that gives other results than this IIR threshold. So we chose to just pick the default setting in the software program, which was an IIR of 1.2, and that cutoff value is being used in a lot more papers. But I acknowledge that the cutoff values and also the post-processing strategy needs to be researched a lot more than it is today.
Okay, so thank you very much again for the great discussion and I think these were really two interesting papers. Matthias, do you want to tell us what the next CMR Journal Club is about? You're still muted.
Thank you. Thank you, Bettina. And also thanks to the presenters for the interesting presentations. So the next CMR Journal Club, because of the SCMR, we will not have it as usual in the first week, and not in the second either, because the second week will be the week of the JCMR Journal Club. So the next CMR Journal Club will be on February 16, but we will announce it before, and there will be 2 papers on a topic that I think is undergoing some changes in our understanding, and this is mitral valve prolapse. And there is a very provocative, but also very interesting paper looking at several features of mitral valve prolapse as assessed by CMR. That's from the group around Javier Sanz and Boria Ibanez, and with Valentin Fuster and Angelica Romero Daza is the first author. And then we also have a paper from King's College team with Caroline van der Heining presenting on dark blood late gadolinium enhancement in patients with mitral valve prolapse, looking at infarcts or scarring in these. So very interesting. Hope to see you all on February 16 then. That's all from my side today. Thank you.
Thank you, Matthias. I'm looking forward to these 2 papers as well. So yeah, thank you again to the 2 presenters, and to you all for joining today and for the lively discussion. Hope to see you the next time on February 16, and stay healthy. Bye-bye. Have a nice evening.
Bye-bye.